Hi guys, so today we will be exploring through life science class, which is all about microbes, about cells, about their genetics, and about how it can be applied in the industry of engineering. So let's begin with our first chapter. We have here microbial genetics. So microbial genetics is the study of genes and basically instructions inside of the cells. It is made of DNA, controls how cell works and what protein it makes. So we have the nucleic acid, and nucleic acid, either we have DNA or RNA. DNA stores information and RNA helps to use that information. So the difference between DNA is RNA is that the DNA, as you can see from the picture, it is double-stranded and RNA is single-stranded. DNA has deoxyribose sugar and RNA has ribose sugar. The main difference in genetic code is that DNA has thymine. This T you can see, it actually opens as thymine. And the RNA has uracil, but all rest, like we have here adenine, cytosine, guanine, and here as well, they are all the same. And the DNA has more stable, as deoxyribose sugar is considered to be more stable sugar, but RNA, the ribose sugar, is considered to be less stable sugar. And you can see here we have a more clear picture, and this is the chemical structure of thymine, cytosine, guanine, adenine, uracil. So this only exists in RNA, as we've mentioned, you can see here, and the thymine only exists in DNA. Okay, so let's explore the structure of DNA. Each DNA unit is called a nucleotide. What is nucleotide? Basically, it's when we have sugar, phosphate, and base. And this is called a nucleotide. But if we consider only sugar and phosphate, that is a backbone of DNA. And the direction of DNA is from 5 to 3. From 5 end to 3 end. And you can see here these ones, they are actually attached by the hydrogen bonds, and these are actually base pairs. And as I've mentioned, this is the sugar phosphate, this is the backbone, and this all together, it's a nucleotide. DNA in prokaryotes and eukaryotes can be a little bit different. In prokaryotes, we have got no nucleus, one circular chromosome, no histones, so and we have plasmids, like we have let's say this is a DNA and we have additional DNA, the small additional storage, that would be called plasmid. But in eukaryotes, the DNA is actually so long that DNA can be like many meters, and we cannot simply fit it. So we have a thing called histones, like this perhaps, and the DNA wraps around it, if you can imagine. Okay, moving on, we have purines and pyrimidines. Purines are large bases and pyrimidines are small bases. Usually purine pairs with pyrimidine and they stay together via hydrogen bonds. The large base includes adenine and guanine, and the small base includes cytosine, thymine, and uracil. So, and this purine and pyrimidine stay together via hydrogen bonds, but sugar and phosphate stay together via phosphodiester bonds. You can see here, you see the as we've mentioned, the purine is the bigger one here, and the pyrimidine is the smaller one, and they are attached, as you can see here, they are attached by hydrogen bonds. And on the 5 end we usually have a phosphate end, and in the 3 end we usually have an OH. And prokaryotes, they have continuous genes, but eukaryotes, they have exons and introns. So I'm going to explain further what is exon and intron. Usually introns will be removed later. So then we have a concept of central dogma. So DNA turns into RNA and RNA turns into protein. That is called central dogma. And first we have replication, where DNA makes copy of itself, because DNA is kind of untouchable, so it usually makes a copy of itself so that it is not harmed or anything. And then we have transcription, where DNA is transcribed into RNA. And then we have translation. Since RNA has a different language and protein has different language, we say it as translation. Their RNA turns into a protein. So we have three types of RNA. We have messenger RNA, we have transfer RNA, and we have ribosome RNA. So messenger RNA carries the message, the transfer RNA brings amino acids, and the ribosome RNA makes proteins. And proteins are made of amino acids. So we have DNA, it transcribes, it's like copies itself, and it transcribes into mRNA, and then it translates into protein. And you can see these bonds here in the proteins. How are they even kept together? So we have an enzyme called chaperones. They usually help the process. And of course we have here hydrogen bonds, we have van der Waals forces, we have different kinds of bonds that keep them together. Let's talk now more in details. Let's talk about DNA replication. So in DNA replication, DNA copies itself. So before that there was even process of replication where DNA copied itself, and it happens before the cell divides. So two strands, they separate, and each old strand becomes new, becomes a template, and new matching strands are made. Okay, let's say like we have a cell division, and in the cell division we have one DNA, right? But when we divide, each of them is supposed to have its own DNA, right? So the two strands, we have a DNA, let's say, and the two strands, they separate, and then each old strand which has been separated becomes a template, and then the matching strand according that template is being made. And that's why DNA is actually called semi-conservative, because it always consists of one old strand and one new strand. DNA opens at specific locations, forming the Y shape. So let's say we cannot simply where it starts to open. Let's say we have our DNA like this, and at some point it needs to open, let's say from here. And when it's opened, the two strands, as we've discussed, they separate, and it turns into Y shape, which is called replication fork. And it forms into two directions. So 3 to 5 is actually forming the Okazaki fragments, because it is against the original direction where DNA is usually moves. But 5 to 3 we just have normal movement, but since it cannot move properly from 3 to 5, it usually moves by forming these small parts, which are called Okazaki fragments. And then we have enzymes in DNA replication, and we have quite a lot of enzymes in DNA replication. So we have first enzyme, it is helicase. So as I've mentioned before, if we go back, they are together by hydrogen bonds here, right? They are together by hydrogen bonds, and we need an enzyme to break down those hydrogen bonds. So the helicase, we have to remember, helicase open DNA double helix and breaks hydrogen bonds. And then we have single-stranded binding proteins. They are usually called SSB proteins as well, and their separated strands might join again because they tend to join again. So they kind of hold DNA open and do not let them join. Then we have DNA gyrase, or we can also call it topoisomerase. When helicase opens DNA, DNA ahead is a DNA which goes ahead of it is kind of twisted and stressed, and it removes twist stress. So we can also remember in our mind this enzyme, we can remember as like anti-stress enzyme, kind of helps to release the stress. Then we have primase, which is a starting piece. Then we have RNA primer, short RNA sequence, let's say 5 to 15 nucleotides. The RNA primer consists of this 5 to 15 nucleotides. It is temporary and it is just to start DNA synthesis. Later, we remove it. Then we have DNA polymerase 3. It's just shortly I write pol. It adds DNA nucleotides to 3 end of growing chain, and it proofreads 3 to 5. So as we open and we are forming the new DNA, this DNA polymerase 3 is going to come and add DNA nucleotides to 3 end of growing chain. And usually it is going to be adenine, thymine, guanine, or cytosine. And it also proofreads 3 to 5 because there might be any mistakes, so it usually comes back for proofreading. DNA pol 1 removes RNA primers and replaces with DNA. And then we have DNA ligase. On lagging strand, DNA is in pieces. So let's say, as I've mentioned before, on replication fork, we have the Okazaki fragment, and just like this, right? So the ones that have Okazaki fragment, this is called lagging strand, and this is called leading strand. So the DNA is in pieces, and the ligase kind of helps them to join together. Okay, so this is it for the first process, which was DNA replication. These are all the enzymes that help us. This was all just a process of DNA copying itself. And the next process, we have transcription, where we turn DNA into RNA. So the RNA produced can be, as we have mentioned before, it can be a messenger RNA, tRNA, or ribosomal RNA. But the most important one, we have messenger RNA. So one strand is used to make RNA, and that is template strand. So we make RNA with the help of template strand, and it's usually 3 to 5, and RNA polymerase turns it into 5 to 3. So RNA is being built from 5 to 3. And we have an enzyme here, RNA polymerase. It's that enzyme that makes RNA, but you can get a bit of confused because here we had also DNA polymerase. We have DNA polymerase 3. By the way, just an interesting fact, don't pay attention to numbers. Even though it's polymerase 3, it comes before polymerase 1. Anyways, so we have DNA polymerase and we have RNA polymerase, and we gotta know their difference. So DNA polymerase makes DNA and RNA polymerase makes RNA. It needs primer, but this one doesn't need primer, and it used the NTPS. So it's, I believe, just a DNA nucleotide sequence, and the second one is RNA nucleotide sequence. It proofreads well. DNA polymerase proofreads well because we are gonna use it temporary, but RNA is, we are gonna use it like forever. But RNA polymerase is temporary, so it's got little or no proofreading. So what are the stages of transcription? We have initiation. First, RNA polymerase binds to promoter, which is basically a starter before the gene. Then we have elongation. RNA polymerase moves along DNA template and adds complementary RNA nucleotides. And RNA chain can grow 5 to 3N. As I've mentioned, DNA grows 3 to 5, RNA grows 5 to 3. And as you know, adenine binds with thymine, cytosine binds with guanine, and uracil binds with adenine as well. So we can see here we can replace, because usually the RNA is going to include uracil. So what are we going to do? So we just replace them, like thymine replaced with adenine, the one which pairs adenine and uracil, cytosine and guanine, and again guanine and cytosine. And lastly, we have termination. When RNA polymerase reaches a terminator sequence, and we have some methods which we can terminate with. So we have stem loop structure, where RNA folds back on itself and forms kind of like loop, and it makes RNA polymerase pause, and weak uracil and adenine bonds cause RNA to detach. And then we have ROPE. It's a protein row dependent termination. The row binds to new RNA and moves along it. When RNA polymerase pauses, row catches up and helps RNA to separate from DNA. So let's talk about structure of mRNA. So we have a leader sequence, coding sequence, and trailer sequence. Leader sequence is before coding region. Then we have the coding sequence, where we have instructions for amino acids, and trailer sequence. Usually you don't need to know all of the coding sequence. I can show you, like here we have all coding sequence in biology. Like we have this, we have this bases, they're called codons. We don't need to memorize all, but it's great to memorize the stop codons, which are UAA, UAG, UGA, and start codon, which is AUG, codes for methionine. And lastly, we have translation. mRNA message is decoded to build a protein. So we have RNA language, they consist of nucleotide, and protein language, it consists of amino acids. That's why when we turn RNA into protein, that is called translation, because they as I've mentioned before, they've got different languages. So mRNA carries the message and has codons. tRNA brings amino acids to ribosome. Each tRNA has amino acid, anticodon, and anticodon, which pairs with mRNA codon. So what is the process? mRNA is like a recipe. Ribosome reads it. tRNA brings correct amino acids. Peptide bonds join amino acids. Protein grows. Stop codon, and then protein is released. So this is basically the complete summary. And we also have peptidyl transferase catalyzes formation of peptide bonds. So here we can also, we use the enzyme peptidyl transferase is used. So this is all about, we have basically central dogma. I want to show you in illustration as well. Central dogma of molecular biology. Okay, so first we have transcription from DNA to RNA, and we translate it into the protein. And here, as we've discussed, difference between DNA polymerase and RNA polymerase. DNA polymerase is basically responsible for replication. The RNA polymerase responsible for transcription. And I just want to see if we have more detailed. Okay, you see here, first we have replication. Okay, this is more detailed picture. First we have replication, where DNA duplicates itself. You see we have two DNAs from one, semi-conservative, right? And then you can see here basically the strands, they separate and we kind of form a Y structure. And all of the enzymes that we have discussed, we have like helicase, lipase, and so on, they are all here. And in RNA polymerase, and then it becomes here that you can see messenger RNA, and that messenger RNA is being transcribed into protein at a specific sequence, right? Like transfer RNA brings the amino acids to the ribosome, and they're all getting transcribed into protein. And here at the transcription, yes, you can see how it's being separated. So I hope this process is more or less clear to you. Yeah, thank you for listening, and if you have any question or correction that you can try to make, you are free to tell me. Just let's see this one as well. So we have transcription, the process of transferring genetic information from DNA to complementary strands of RNA. Okay, in translation, as you can see, each codon codes for a different code. Like as we've discussed, start codon like codes for methionine. There are different codes, but not every lecturer wants you to memorize each codon. And in translation, the process of transferring genetic information from RNA to a sequence of amino acids in protein. The sequence of mRNA is read as a series of nucleotide triplets called codons, and each codon specifies one amino acid. So when we translate, this language is translated basically to protein language. And lastly, the protein folds, as I've mentioned before, like the chaperones keep them together. Polypeptide chains undergo a series of folding to form functional proteins. So folding is important. Okay, I think that's it for this lesson. Thank you.